Because you're last.
Marcy's probably and Jack's family are like BFFs.
Okay, let's get started. So lab, there are some rules. And I want to go through the rules. They are in the front of your lab book. I don't know if you've had a chance to look through those or not yet, but some of them I want to talk about even though they're written down. No open food or drink in the lab. Okay, so I'm going to have to get a closed container for my water too. You can bring in closed containers if you want to, water bottles and things, but no food because the chance of contamination and you getting sick from stuff that's in the lab is just not that high, but we just don't want to take that risk. Okay. Please make sure that you practice cleanliness at all times in the lab, and that includes both benchtop and personal. And so you really should get in the habit of when you come in, washing your hands. There will always be antibacterial soap at the end of both benches right here. And particularly when you leave, make sure that you wash your hands, okay? And most people don't wash their hands nearly long enough to really get good coverage. The old kind of rule of thumb that I've heard is that you have to hum Twinkle, Twinkle, Little Star twice to really get a good hand washing done. Most people don't do it that long. But make sure that you practice cleanliness washing your hands. When you come in for lab day, you should use this Clorox cleanup and a sponge to wash your bench area. I don't know if you've noticed on the outside door there, but we did some street plates on the surface of washed and unwashed lab benches a few years ago. And the plates that we streaked from unwashed tabletops had all this crud going on it, and the washed ones were really pretty good. Okay, so if you're particularly working with your unknown, you don't want to get contamination picked up from your tabletop. Spills. If you spill something, and the most likely thing you will spill would be a broth culture, then we need to do some cleanup. We're going to use 3 types of media in this class. We will use auger plates. We will use auger slants, which is just auger in a sterilized tube. And then when it was cooling, we laid it down on the side so it's got a good surface area for streaking microbial growth. And then broth tubes. Okay. All of these 2 tubes have push-down caps. They're not screw-on. And so if you lay these down on their side like this, okay, this is sterile, don't worry about it. Okay. Then it's going to start dripping all over. Okay, so you need to keep these upright. So if you do spill a liquid culture, let me know. And then we can either use, if it's minor, probably just the Clorox cleanup. So spray a little bit of that Clorox stuff on there, go get a paper towel. And then the paper towel should be put in an autoclave bag, because then we'll sterilize these as they fill up so they don't contaminate anything. If it's a really bad spill where we've got something that's kind of nasty you're working with and there's a big pool of liquid, let me know. We've got some of this powder up here called Biosorb, and you sprinkle that all over it and it kind of soaks up the liquid, and it's got some good strong antibacterial stuff in the powder, and then we can scoop it up and sweep it up and throw it in the autoclave bag. When you're working with cultures, after you inoculate them, you will have to put them in an incubator to let them grow. After they've been in the incubator for no more than 48 hours, because otherwise they start drying out in there, then if you want to save them, they go in the refrigerator. Eventually, we're going to have to dispose of them. Okay? Disposal will be in the hood over here, unless it's an auger plate. Auger plates can be put directly in these autoclave bags here. Do not put test tubes in the autoclave bags. Okay? There will be test tube racks over here, and those can be used to put the auger slants in. I'm sorry, the broth tubes in. And then we'll have these little white baskets right here, which I typically will lay on their side so you can kind of lay the slants down there. If you put the broth tubes in here and then lay them down, they're going to start leaking all out, okay? So So try to remember to put the slants in the baskets, and then the broad tubes will be in the test tube racks. And we have work study students who will come in and take care of those and autoclave them and clean them up and things like that. So you don't have to worry about that. Okay? What I'm going to have you do today is each pair of people, and what we'll do for pairs right now is just 2, 2, 2, 2, 2, 2, and then we'll have a group of 3 right here. Each pair of people grab a test tube rack from under the water bath by where Hannah's sitting right there. Put a little piece of tape on there, put your name on it, and then stake out a spot in this refrigerator to put your test tube rack, okay? And this is where you will then keep cultures that you want to keep. Your unknown, for example, or stuff that you might want to check later. But this is where you'll be your temporary storage until you dispose of things, okay? We will be using Bunsen burners a fair amount. Make sure that when you finish using your Bunsen burner, it is tightly turned off. If you ever come into the lab and you smell gas, please let someone know. Natalie's around, Chris is around, I'm around. If it's not too bad, you might come in and say, real quickly check these and make sure that they're shut down. But probably the safest thing to do if you really smell gas is hit this red button right here. And that will shut off the gas to all of the gas jets. And then let somebody know, and we'll come in and figure out which one's on. This little key here can then just be flipped and now turn them all back on again. But please, if you ever smell gas, if you ever even do something about it, let somebody know. Okay. If you have any cuts or abrasions on your hands, you really need to let me know so we can see that they're properly bandaged. I will have gloves down here, so particularly if you have cuts or abrasions, you should put on a pair of gloves. Probably you're going to want to use gloves when we do some of the staining procedures, because otherwise your hands get all green or purple or whatever. And as I did indicate, there are lab coats in the back there. As you walk around the corner, there's several hanging up on pegs. You can pick out a lab coat, and each person will have a drawer, so you could stuff your lab coat in your drawer if you want to, or you can hang it back there. I really don't care, that's fine. Told you about spills already, told you about culture disposals. Never pour a liquid culture down the drain. Okay, it's always got to be sterilized first. Talked about gas already. Each one of you will be assigned a microscope, and you can just come over here and pick them out at random. In fact, one of the things you might want to do today is just identify one of these. You don't even really have to take it down. What I would do is just put a piece of tape on here and put your name on it. That way you'll make sure you always have the same microscope. It's very important to keep your microscopes clean. We're going to be using a lot of oil immersion in this class. And so at the end of every period, you need to make sure you take lens paper and wipe off all the lenses, including the oil lenses. It's also standard practice when you store the microscope to have the stage in the full down position, light turned off, and the scanning objective should be the one that's in viewing position. Okay? So take care of your microscopes. When you carry them, they're heavy. Carry them with 2 hands. There is a little hand hold right on the back right here. And then the second hand should always be under the base. Okay? If you drop one of these things, I mean, 1,000 buckets. So please treat them with respect. We just had them serviced, and so they've all been checked out. They should all be in good shape. But make sure you keep them clean. Oculars tend to get smudged up with grease and things from eyebrows, and so use your lens paper to clean off oculars, and also lens paper to clean off the objective lenses. There are 2 types of paper that you will have in your drawers. We'll go through your drawers in a second. But lens paper and bibulous paper are 2 different beasts. Lens paper is the kind of stuff that's kind of like tissue paper almost. Bibulous paper is thicker paper, it's blotting paper, and it's used for slide preparation. So don't try to clean your lenses using bibulous tape. Make sure to use the lens paper. Everybody will have 3 drawers. The top drawer we'll go through in a second to make sure you have all your supplies. The middle drawer has some paper in there, some lab mat paper, and that's where you'll store any slides that you want to keep that you've made. I would recommend you keep all of the slides that you make because you will likely want to review them prior to the lab exam. Bottom drawer has Bunsen burners and staining trays in them. We'll make sure you all have those in a second. If you do want to bring things in here and leave them in here, like your lab manuals, you can. If you would like to have your drawer locked for security, let me know, and there are keys available for the top drawers. I've really never had anything disappear here from unlocked drawers. We don't have to share this lab with anybody this semester, and so the lab is open all day. So if you do want to come in and work on lab stuff outside of normal lab period, you're more than welcome to. As a matter of fact, probably later in the semester, it will probably become fairly common for you to have to do that. There will be some days when we will finish early, and that's fine, because there will be other days when you're going to have to do some more extended work, particularly when you're working on your unknown. So I never feel bad if you get out a little bit early sometime because you more than make up for it. Keep your lab space clean. The tabletops tend to get a little bit cluttered, particularly when we start getting some of the staining reagents out here on the top of the table. Particularly since we have our lecture sessions in here as well, it's important to keep this clean. Keep your tabletops cleaned off with the Clorox cleanup so you don't pick up any microbial contamination and make yourself sick. And no, I don't really know ever of an instance where in the 30 plus years I've been teaching this that someone has gotten sick from an organism we've been working with in microbiology. Okay. But it's important to treat these organisms with care. Some of them are potentially pathogenic, okay, particularly if your immune system tends to be a little bit down. If you develop kind of a habit of, well, I'll treat some things carefully and other things because they're fairly common and not bad, then I don't have to worry about them, you get into sloppy habits. Treat everything as if it were a pathogen, and then if you treat it with that level of respect, you'll be good. Have to read the lab materials before coming to lab. I reserve the right to give you pop quizzes if I think that you're abusing that. But ultimately, what it really hurts is you, because if you haven't read the lab ahead of time, then you're not going to finish on time. Okay? I don't give any real pre-lab introduction. You are juniors and seniors, and you are able now to take responsibility for that. So I'm going to assume that you have read your lab material, and then at the very most, when I come in and say, here's where the stuff is, I've got this stuff here, maybe occasionally we'll have a substitution of an organism. Sometimes I'll organize you in a little bit different groups to kind of save media. So in some things you're going to be working in pairs. Other times I have groups of 3 or 4 that work together and share inoculating known control cultures. So I'll make sure that that is well spelled out before each lab section. Particularly when you're working on your unknown, if you find that we're starting to run short of materials, you have to let us know. Typically, we will have a set of slants and broth tubes just sitting out here that you're able to use for any culture transfers. Auger plates tend to dry out a little bit, and so they will typically be in the bottom shelves of this refrigerator, the bottom drawers, the pull-out drawers. So if you pull those out, there should be auger plates in there. If we get to a point where you see things are getting low, and particularly if you know you're going to need something a couple days down the road, then you can just make a little list up here in the corner for the lab assistants, and I'll make sure they check that. So if you start running out of slants, just write slants up there, or plates or something like that. Let us know. If you start running out of staining reagents, crystal violet for the gram stain, iodine for the gram stain, then again, put a little note up there and indicate, we're starting to run out of some of the things. First lab, you will have to make your own media just so you know how to do it and where it comes from. And then for the rest of the semester, we have lab assistants that will prepare all the media. So that'll make your life easier. Anything that needs to be refrigerated should be put in your rack and carefully labeled. Occasionally, I will ask you to clean out the refrigerators, usually right around the lab exam time, because they tend to get pretty full, and people probably save things too much, particularly related to your unknown. So we do have a limited amount of space available in the refrigerator, so be respectful of your classmates there. Lab coats or grunchy clothing on lab days usually. Lab's not going to be open on the weekends or evenings unless we run into some kind of a crunch where you say we really need some more lab time. If that's the case, then I can probably talk if I can schedule it. One of our work study students to come in and just be a lab proctor. But typically, because the lab's open pretty much 8 to 5 every day, that's not a big problem. So any questions about these?
Do we all have our own unknown, or just
You
like
all have your own unknown, yep,
Okay.
yep. So you will work in some kind of group to do the control known organisms, but everybody's responsible for your own unknown. And last year I started something new, which worked out really well, I think. People were required to document all of the test results of their unknowns electronically. We will have a microscope with a camera set out over here where you can take pictures of the slides that you make. If you have an iPhone or an iPad, you can just plug that right into the camera. If you don't have one of those, we have one downstairs in Natalie's office that you can use. And I think it works with, I know it works with the 5 and the 5S, probably works with the 4S as well. But then you can just email those to yourself, and then I ask you to put those into a PowerPoint, and that is part of your lab write-up, is all of the test results. It's great because in the past people would go, wow, I wrote down this test result and I put positive. And I'm having trouble keying it now, and I'm going, was that really positive? Well, if you can go back to your picture of it, you can go, that wasn't positive. Then it can be really helpful. And it also then gives me a good way to check if you've made a mistake in your keying. If that mistake was based upon an honest interpretation of a result that was wrong, then I will take that into account when I do your grading. Because bacteria mutate. And sometimes your strain will be a mutant strain. And Bergey's manual will say, it is supposed to be positive for this test. And if that's a key couplet in a key, and you say, well, it was positive, I go this direction, you could end up with the quote-unquote wrong answer based upon legitimate results. And so I can go back then and look at your results and say, yeah, they were right, that was positive. And therefore that legitimately did lead them to this identification. So you can, it's not uncommon for people to get full credit for their unknown write-up, and they might come up with a different species than I thought I assigned them. Potentially even a different genus, although that's unlikely. Okay, so that documentation really makes it easier for me to grade fairly, but it also gives you then a better way to evaluate your results. You also do have to keep a notebook of your unknown results and your class results. I don't think on the syllabus I am grading that. That's really for your purposes. Each lab has a set of lab questions at the end. I highly recommend that you answer those lab questions, but I'm not going to collect those and grade those. But many of those lab questions are the kind of questions you will see on the lab exam. Okay. If you've looked at the syllabus, lab is a pretty big component of the grade of this course. There are 2 100-point lab exams. Your unknown is 100, 150? 150, I think, yeah. And so lab really is about half the grade. So that's very important. The atmosphere in this lab tends to be fairly informal, and I think that's fine. Those of you that know me, I like to play music during lab. I think it just makes things go faster. Some interesting dynamics oftentimes develop during lab. A fair amount of bantering and kibitzing that goes on, particularly between the little lab groups or partners. Sometimes people get like old married couples after a while, and they're John and each other about something they've done in lab. But I think that because this lab is so hands-on and that it's so geared toward a project that you have some investment in, things tend to go pretty fast, and people seem to enjoy the lab and think it's a very important part of the course. And so people are moving around a lot, and if you have something interesting on one of your slides, for example, please let the rest of us know. Sometimes in some of these staining techniques that we do, only about 60 or 70% of the slides really work out very well. So sometimes I'll have to ask people, if you've got a good result, let me know so other people can look at it. Sometimes you'll have to repeat results because not all the tests work out the best the first time. Okay, if you would open your top drawer, please, and let's make sure you've got your materials. If you don't have materials, let me know. So each person should have their own drawer. So actually, in this row, your drawers are to your left. So that's Hope's. Okay, in the top drawer, you should have some kind of a test tube rack. There should either be a half rack or a full rack. If you don't have one, let me know. Okay. Do a half one here. Oh my goodness. Back to this one, right? And if you see some kind of miscellaneous junk in your drawer that I don't tell you should be there, get rid of it. Okay, everybody got a test tube rack? Okay, probably at least every other person, if not every person, should have a box of Kim wipes in there. Those are the green boxes of kind of tissue-like material. So if every other person has one, that's probably fine. Everybody needs a sponge. And if your sponge is looking kind of nasty, we have some replacement sponge here. Some of them. That one's actually not too bad, but we'll give you this one anyway. They tend to get kind of black over time when you use the Clorox cleanup to wipe these things off. So if you've got, there's a nasty sponge. That calls. Yeah, you're just. You okay with living with yours? You want to got something down. You guys not have anything down there? I thought the year-old ones here.
Good or bad, I don't know.
Yeah, that's true.
I know, I didn't.
Don't give it to me, I don't want it.
Definitely.
Grab one out of your hand.
I get scared. Like if it's not one of like.
Okay, everybody should have a test tube holder. We use these for grabbing slides when we clean slides. So anybody missing a test tube holder? Yeah, 4. Well, if you have extras of stuff, give them to me. Somebody was a hoarder. Everybody got a test tube holder? You do.
You got all sorts of problems.
Sharpie? Everybody got a Sharpie? Again, if you have extras of anything, toss it to me. We'll distribute them.
Which one do you want?
Sharpies.
You figure it out.
You got extras over there?
I have one.
With your Sharpies, if spines are right, you should write on the bottom of the Petri plate rather than the lid, because the lids can come off.
I throw it at you.
Where's my
You act like you're paying attention.
When you write on test tubes, we would prefer that you write on the side of the test tube rather than the cap. The caps are kind of a pain to clean. And sometimes we dispose of these test tubes anyway. Okay, everybody should have an inoculating loop. I think that is the green one right there. And you might want to check and see and make sure your loop is intact. Sometimes the loop will split because it gets heated too much. Everybody got a functioning inoculating loop. And then you should also have a straight needle. I think those are the blue-handled ones. You need needle? Anybody have an extra needle, or do we
Yes.
see it? Okay.
Oh,
Oh,
I'm just chatting.
okay. You should have a booklet of lens paper. It says lens paper on it. Okay, if you need a. Oops. Have another one. Okay, nebulous paper. And if it looks a little nasty, I mean, you can actually reuse it again. Now, sometimes when they get all really colored like this, you want to just toss them. But many times the books will have some used pages and some unused pages. If you ever need more nebulous paper or lens paper, it's up here. So anybody have some missing booklet or? Okay. Okay, matches. Do we need matches? Those? How many there are there? Okay, everybody should have some kind of a little slide box that might be plastic, and it might be just paper, cardboard. And in that should be 2 depression slides. Anybody sure of a depression slide?
Okay, we have more.
If you have more, give it to me, because I think Jeremy short one. You only have one. Do you have an extra one? And then you should have 3 or 4 glass cover slips. What I'm going to do is I'm just going to pass a box down each table. And if you need to stock up a little bit, do that. Good one. Everybody should have a bottle of immersion oil. Some of you may have multiples.
How many are we supposed to have for,
2 or 3.
okay, well, you can't take it.
Okay, I think there's some more. Are there some more in that one? Just finish your. You got extra? Yeah. This is immersion oil. Will either be in little glass bottles or plastic bottles. And if you're getting low, we can top them off. Actually, I'll leave this at the end here. If you want to top it off, you can.
I don't have that much.
No immersion oil? Okay, let's. Does anybody have an extra immersion? I do. Do you? Well, okay. Yeah. And then you've got a blue box, and I'm not sure we really even need this stuff, to be honest with you, but the blue box typically contains an eyedropper, forceps, dissecting needle, razor blade, and ruler. I don't know if we've used the blue boxes very much. If there's something you're missing, you really want to have, you know. Anybody else need oil to top off your oil? Anything else that's in your drawer that I didn't call out, you can toss or give to me and I'll put back on supplies. One of the things you will use a lot of will be microscope slides. And I will try to remember to have microscope slides out all the time. It's probably over there about where the plants are. But there's a drawer down here that has boxes of microscope slides, so if you need more slides. So basically, micromaterials are oftentimes over these drawers over here. Don't feel bashful about coming in and stocking the supply of something you need. If you need it. Everybody have the paper in your second drawer, lap pad. If you have slides in there, they can be thrown in the autoclave bag if somebody left slides over for you. Okay, your bottom drawer should have a tray with a wire rack that fits over the top. There should be one per drawer, so some of you have more than one already. And then there should be a Bunsen burner. People probably have extras. It's probably not a bad idea. There are 2 kinds of Bunsen burners. There are the new shiny big ones and the old ones. And they have different uses. So probably it'd be a good idea if kind of every other drawer had one of the different kinds of Bunsen burners. So if you can swap around a little bit.
Yes.
The new ones, particularly the first few weeks of class, if everybody's got one of those new ones on, they're really hot and they really pour out the heat. So sometimes we'll ask you to use the old ones when possible. So everybody have Bunsen burner? Everybody got a staining tray and rack? Have an extra Bunsen burner? Oh, okay. 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 Blood pressure slide?
Yep.
That's disgusting.
What is it?
Okay, anybody missing anything? You may have an extra tray? Do you have a wire rack for it? Yeah, you're just missing a tray.
The little cup, yes.
Things move around during the semester.
I don't know if
Oh, we just, I just.
Oh, okay. Oh, can I have that?
Are there cover slips still on that one, Ken?
That was it, please.
It's empty? Okay.
You can hear the description. I'm quite all right with that. Sure, I see next to you.
Since we don't have, and if there's anything you don't know, like these miscellaneous things right here, these will keep your dishes to be hot. But if there's something you don't know, just stick it over there by where my slides are, and I'll take care of it. When we do staining, since we don't have in sink in the benches right here, you'll just be staining on top of those wire racks, and stuff will drip down into the staining tray. Unless I tell you otherwise, we can dispose of the stains down the drain. They're just dyes. And so I will make sure I let you know. Typically, if we can't dispose of something down the drain, we'll do it under the hood. There's xylene rinse we have to do as one of the staining procedures. But make sure your staining trays are washed out before you get back. So everybody equipped? So like I say, if you want to keep things in your drawer, that is your drawer now. If you want to keep lab manuals and stuff, then that's fine. If you do want a key, let me know. Otherwise, shut the door. So next week we will do lab number 1, which is several things. It is an introduction to microscopy, particularly using oil immersion lens. We will learn how to standardize your ocular micrometer so you can then measure microorganisms. That's one that sometimes gives people a little bit of problem to figure out. We will look at some representative morphologies of bacteria, Bacillus cocci, Spirillum. We will do a little exercise on cleanliness with hand washing and clean and dirty countertops. And you will make some media. Typically, what we'll probably do is split things up, so one table will start working with media first, the other will start with microscopy, so we're not all standing in line waiting for available materials. Okay. So make sure you read your lab ahead of time, and we're ready to go next Wednesday. Oh, yes, I'll put some tapes over here. And if you would get a test tube rack for your group,
I have one. But in this basket, it's like. Okay, my pencil happens. No, actually, these two are neat. Mine last year was like, so today I have
somebody do my tape.
Is it white or regular?
Well, I've got several rolls of it.
Does that have to take in my drawer?
There's some in your drawer?
Yeah.
There was? Okay.
Yeah.
Is there much there? If you would use that then, just tear off a little piece to put on your test tube rack and another piece to put on the back of your microscope. I'll see if I can find my other box of it. One other thing, particularly as it starts getting a little bit cool, if you could try to put coats and jackets out on the coat rack out there when you come into lab, so they're not hanging off the back of stools. And backpacks and things, if you can kind of keep out of the aisles. We have the luxury this time of this aisle being open, so we can actually get down this aisle. This semester, last semester, it was all filled up. So you've got a little bit more freedom of flow around here. So we got out early today. Yeah.
I just want to introduce and if you have any like concerns
certifications. I don't, but I have really bad eczema.
Oh, then take gloves. I mean,
Okay.
yes.
Because sometimes they'll open up, sometimes
Oh,
they won't.
yes.
I take medicine for it, so.
Absolutely, yes. Just,
Thanks.
and let me know if we start. What size do you wear normally?
Small or medium. I'd say, I don't know, what do
Should
you?
we just grab you a box and you can put in your drawer?
Sure.
Okay.
Just eczema sucks.
After we label our stuff, are we good to go? Yeah.
Hi, Allison. Love you.
Sorry, Zoe. My grandparents shut it off.
It's floating around someplace. I can't find my box of tape.
I'm gonna put mine in there while I'm.
Somebody have a tape?
Thank you.
Oh, it's over by the Steal this? You guys using it? Just pull off a couple pieces and I'll pass it around.
You can't go on.
Yeah,